Once again, good morning to all of you. Today we are to continue the next section of antihistaminic agent, that is S2 receptor blocker, and the continuation part relative to the gastric inhibition, gastric acid secretion inhibitors, that is proton pump inhibitor. Today we are to discuss S2 blockers and PPI, presented by my Dr. Vivek Panchway. Here, S2 antagonist, nothing but antagonist means the opposes the action of S2 receptors. The clinically S2 antagonist is used in the treatment of acid-related gastrointestinal conditions like gastric secretion, acid secretions, acidity-like conditions. This S2 antagonists are used clinically. Specifically, these indications may include peptic ulcer disease, GERD, and dyspepsia. In this S2 antagonist, cimetidine is the prototypical member of the S2 antagonist. Further, whatever the development in S2 blockers or S2 antagonist, cimetidine is the basic moiety, or the cimetidine, whatever the replacement, modification, QACR studies, the further development of the newer molecules are focusing on the basic structure of the cimetidines. So, for the better tolerability profiles, apart from cimetidines, ranitidine, famotidine, and nizatidine, these are the S2 blockers are developed in this segment, S2 blockers. The general structure for S2 receptor blockers, that is imidazole moiety. Here are substitutions, that is alkyl substitutions. There is a chain, is a usually butyl-like chains or isosteric replacement of butyl group with thioether linkage chain present in the S2 antagonist. And this is a terminal basic nitrogen should be present in the structure. These are the basic structural requirement for S2 antagonists. On imidazole ring systems, presence of alkyl chains and presence of the chain in between terminal nitrogen and imidazole rings. As per syllabus is concerned, the S2 antagonists are cimetidines. The structure of cimetidine is imidazoles, is a basic moiety present. The alkyl group is methyl groups, and here the linkage, butyl-like linkage, that is thioether linkage along with this terminal amine with substitution of this group. This is the structure of cimetidine. Further structural modifications, there is formation of famotidines. Here, if you see here, imidazole moiety present in the cimetidine, whereas in famotidine, the thiazole moiety should be present. The chain which is present is thioether chain, and this is terminal amino group present in the famotidines along with these substitutions. Here, the substitution is different from the cimetidines, and instead of alkyl groups, diamino methyl amino substitution is substituted on second position of thiazoles. This is structure of famotidines. Next, further development, instead of biosteric replacement of imidazole ring with furon rings in ranitidines, same chain should be present in the ranitidines, terminal amino groups, and there is some little bit substitution change in this terminal amino substitutions, whereas this is a substitution present on second positions. This is structure of ranitidines and nizatidines. Similarly, in nizatidines, this is a thiazole ring systems. In thiazole, this dimethyl amino methyl substitutions. This is a chain of thioethers. This is a terminal amino groups, and this substitutions was happen in nizatidine. These are the prototypical examples of cimetidine, that is ranitidine, famotidine, nizatidines. The SAR point of S2 antagonist and structural requirement. The general formula for S2 antagonist is basic heterocyclic groups. The imidazole is a basic. By replacing imidazoles, thiazoles, furons, depend on the isosteric replacement of this ring. This is a basic ring should be present in the structure. Flexible chain should be present along with some aromatic rings if possible, and polar group present in the structures. In SAR point of view, these modifications, these are the results of modification of histamine structures. Here, the imidazole ring of the histamine is not required for competitive antagonism of the histamine at S2 antagonist. Instead of imidazoles, thiazoles, furons also showing better activity. Second point, separation of ring and nitrogen atom group with equivalent 4 carbon chain is necessary for optimum antagonist activity. Means whatever the flexible side chain, minimum is 4 carbon containing or 4 atoms present in the chains, either butyl substitution or thioether substitution is present in the S2 antagonist, which gives optimum antagonist activity. And finally, the terminal nitrogen atoms should be present with polar non-basic substitution for the maximal S2 receptor antagonist. Now we have to move with the mechanism action of antagonist, S2 antagonist. Here, for example, we have to consider the ranitidines. This ranitidine completely block the action of S2 receptors. Once the action of S2 receptor are blocked by this S2 antagonist, the histamine cannot be acted. So this histamine cannot be acted due to blocking of S2 receptor. Ultimately, it decreases the cyclic AMP formations. Once the decreasing cyclic AMP formations also decreases or reduces the gastric acid secretions, and it completely stops the healing of ulcer-like sensation by this ranitidine S2, ranitidine, famotidine, cimetidine, nizatidine. These are the S2 blockers, S2 receptor blocker. This is a basic mechanism action of S2 receptor. Apart from this structural activity relationship, mechanism action of S2 blockers, some examples of S2 antagonists. As per your syllabus concerned, next part is synthetic route for the preparation of cimetidine. This is the structure of cimetidine. This cimetidine was prepared from starting material, react with the two molecules of amino acid formaldehyde. They undergo cyclization, formation of imidazole ring, formation with along with esteric form. These esteric forms which undergo reductions to form the conversion of ester into the alcoholic form. This alcohol react with the amino ethyl. There is a substitution by replacing these water molecules and to form the intermediate. After formation of intermediate, what is the substitutions occur? Polar group substitutions on the terminal nitrogen atoms. Once the substitution is occurs, the further formation of intermediate, and once the polar group substituted on the terminal carbon atoms, they react with the methenamines by replace for this group, sulfur group, which is present in the structure, and to give the formation of cimetidine as a final product of this synthesis. Now, up to this synthetic part, these are the H2 blockers. H2 blockers, nothing but it is one of the antagonists which reduces the gastric acid secretion, near about 60 to 70% gastric acid secretion through this release of histamine H2 receptors. If hyperacidity like conditions, these H2 antagonists are given, in that case, whatever the receptor action of H2 histamine 2 receptors, their action was totally blocked by this H2 antagonists, cimetidine, ranitidine, famotidine, and nizatidine. Now we have to move with the next content, which is related with the gastric acid reductions or hyperacidity like condition was controlled by the inhibitors called as proton pump inhibitors. In this segment, we have to discuss with the parietal cells and their proton pumps. What is the action of parietal cells and what is the proton pump? Then proton pump inhibitor, as per our syllabus is concerned, there are 4 types of proton pump inhibitors. Examples are given: amiprazoles, esomeprazole, pantoprazole, and rabeprazole. These are the inhibitors which inhibit the proton pump, which is involved in the gastric acid secretions. And finally, we have to discuss with the mechanism of inhibition of gastric acid secretions. That is proton pump inhibitions. How the mechanism of action, how the particular proton pump inhibitor acted once enter into the our body. Now we have to move with the study related with the parietal cells and the proton pump. Here, if you see the picture, this is the lumen of the stomachs. Here, this blue color indicating receptors, different receptor involved in the gastric acid productions. Here, in earlier slide, I have to discuss, this is one of the parameter who involved in the gastric acid secretion, that is H2 receptor, muscarinic receptors, and cholinergic receptors. Here, all these secretions was through this yellow color line. This yellow color line indicating proton pump, and this red color is a ion channels, calcium ion channels, for examples. Here, the this color is cannuculus. Here, potassium is present in this cannuculus, and chloride ions. This is an influx and efflux of the potassium ions through these ion channels. Here, if you see this arrow, black color arrow, this indicating the proton pump, H plus K plus pump, that is called as influx of H plus and K plus through this channel, through this pump, that is proton pump, and from this chloride ions and this H plus ions to make the gastric acid juice, that is hydrochloric acids. Here in this proton pump, ATP is converted into ADP through this pump. The proton pump out of the parietal cells and potassium ions back in. So it requires energy provided by hydrolysis of ATP to ADP, catalyzed by ATPase enzyme. So the proton pump name is called as H plus K plus ATPase proton pump. In this segment, parietal cell and proton pump, the chlorine ion depart from a separate ion channel. If you see this line, there is a chloride ion depart through a separate ion channel, and from this chloride ion and H plus ions, they combine and to form the HCl in the cannuculus. Thus, the proton ion exit the parietal cell as a counter ions for the chloride ions and then pumps back in the action. A separate potassium ion channel is used for potassium ion leaving the cells. So this is related with the parietal cell and proton pump. Now we have to move with the inhibitors. Whatever the proton pump actions, they are inhibited. Where at what time the proton pump are given? If the excess acid secretion was produced in our body. In that case, the proton pump inhibitors are given by the physicians. As per your syllabus is concerned, there are four types of the uh, examples for proton pump inhibitor. First example is omeprazole. In this example, there are two types of heterocyclic moiety. This is a benzimidazole structures and this is pyridine structures. And these pyridine and benzimidazole structures are linked with the methyl sulfonyl ring. Here, the yellow coloration, nothing but it's a sulfur atom. If you not seen clearly, I am written here sulfur. Now move with the other examples. Similar, the benzimidazole moiety. In other examples, all the examples having benzimidazole moiety, basic structure, pyridines, and these are the pyridine structures in all the remaining proton pump inhibitors. Benzimidazoles, pyridines, and this benzimidazole and pyridine linked with this methyl sulfonyl group linker. So these are the common part for all proton pump inhibitors. There is a little modification to give the better efficacy of the proton pumps. Here in pantoprazole, difluoromethoxy substitutions on benzimidazoles, whereas in pyridines, the methoxy group substitutions. Whereas we have to see with the omeprazoles, the methoxy substitutions, here methoxy substitution, methyl substitution on pyridines or in benzimidazoles. If you go with the rabeprazole structure, here methyl groups, here sodium is present by replacing these hydrogens of the benzimidazoles, and this is a extension of carbon chain in the methoxy groups of earlier examples, omeprazole and pantoprazole. Here, ethoxy substitutions in lansoprazole and methyl substitutions only. So there is a little modification in the proton pump inhibitor give the better activity. Here, why it is necessary some modifications? Because it controls the proton pump actions for longer times. If you see with this example, rabeprazole generally in general, the physician gives 2 times a day, whereas rabeprazole or pantoprazole given single dose because it acted for near about 18 to 24 hours these drugs. And these proton pump inhibitors act as a product like actions and they activated by strongly in acidic condition found in the canicula of the parietal cells only. If acidic condition environment is required for the action of these proton pump inhibitors. If acidic condition more, these drugs exerted specific activity of this proton pump inhibitors. Now we have to move with the mechanism part of the proton pump inhibitor on the basis of structures. There is structure of omeprazoles. If you see, there is first protonations from gastric acids. They activated. There is a substitution of protons on unpaired electron present on the nitrogens. Then shifting of unpaired electrons from pyridine rings, pyridine nitrogens, and formation of intermediate linkage of 5-member ring system with pyridines. Then abstraction of protons, shifting of electrons from nitrogens from spiro intermediates by loss of proton molecules and to form the breakage of methyl sulfonyl linkage from this benzimidazole and pyridines, and to form the sulfonic acid intermediate formations, followed by loss of water molecules and to form the sulfur containing six-member moiety, that is pyridinium sulfonamide structure formations. And these structures, they bind with the proton pump and to block the action of proton pump. In this way, this is a mechanism action related with the proton pump inhibitor in the form of structure bus mechanism of action of proton pump inhibitors. All of you note down this type of mechanisms. Generally, well, in the examination, university examinations, if you ask the mechanism of action for proton pump inhibitors as a medicinal chemistry, the expected answer for mechanism action, not generally H plus, K plus, ATPase inhibition of proton pump inhibitors. This is the mechanism action you have to give on structure-based mechanism action of proton pumps. Protonations, then cyclizations, formation of spiro intermediate, followed by loss of water molecules, intermediate formation, that is sulfonic acid intermediate formations. By loss of water molecules, there is formation of pyridinium sulfonamide structures, and these structures bind with the proton pump, thiol group of proton pump, and to make the inhibitory action of proton pump. So, up to this part, it is related with the H2 blockers and proton pump inhibitors.